Hello students. In this lecture we are going to discuss about the production of secondary metabolites, that is antibiotics. In previous video we have discussed about the production of primary metabolites like amino acids, vitamins, enzymes, organic acids, etc. Now secondary metabolites. The question arises when organism produce the secondary metabolites. So secondary metabolites are produced at the stationary stage of the bacterial growth, where the nutrients are at the very low amount, or we can say when the organism faces the stress condition, at that time the secondary metabolites are being produced. So the example of secondary metabolite is antibiotics. Now, what is antibiotics? These are the substances derived from the microorganisms or produced from the microorganisms. Or in other terms, we can define antibiotic as a substance produced by the one organism which inhibits the growth of the other organisms. Okay, these are produced at the stationary phase, used for the treatment of the gram positive bacteria, means the disease which are caused by the gram positive bacteria. To treat that disease we use the antibiotics, okay, especially penicillin. Here in this lecture we are going to discuss about the production of penicillin. Now a fact about antibiotic, if we will see just about the penicillin, then penicillin, it is the, we can say it is first naturally occurring antibiotic ever discovered. There are about 50 types of penicillin, like amphoxicillin is there, ampicillin is there, etc, etc. Different types of penicillins are there. Before starting the penicillin, let me tell you more about the antibiotics. Antibiotics can be produced or can be done by the three method. First is natural microbial production, which is done by using fermentation technology. In that example is penicillin. Second is semi synthetic production, which is the post production modification of natural antibiotic. Example is ampicillin. Third is synthetic production, which is completely produced in the synthetic manner. The example is quinol. So this is all about the antibiotics. Now penicillin. Penicillin, it was first developed or first used during the World War II. Massive use was done of penicillin in the World War II. The most important aspects to be considered in the production of penicillin is mass transfer. First, mass transfer. Good transfer of oxygen must be provided into the liquid medium, which is done by the sparger. Sparger, it is located at the bottom of the fermenter. Second is heat transfer. Cooling jackets are provided to maintain the temperature of that fermentation media. Mixing is also important. Mixing is done by the impellers to provide the proper agitation or to provide the proper oxygen transfer. Next important aspect is steam, which is used to keep the reactor running aseptically, means to provide the sterile and aseptic condition. Here the feedback system is being used. Feedback system means here substrate is added in the small amount at various time interval in fermentation, and then consequently volume is being increased. That is the fed batch fermentation. The raw materials which are being used for the production of fermentation is the carbon source. Carbon source, which includes the molasses is been there, or soya meal is there, acetic acid are there, alcohol is there, lactose sugar is there. So these are the raw materials which are being used. Nitrogen source, in the nitrogen source you can use the corn strip liquor. yes, cons triplicar you can use. Here the medium is been given. The medium consists of the fermentation carbohydrate, potassium dihydrogen phosphate, organic nitrogen source, phenyl acetic acid precursor, some edible oils, calcium carbonate which act as the buffer, and pH is maintained at 5.5 to 6 or approx 6.5 level. Okay. द मीडिया फॉर्मुलेशन इफ वी विल सी, अब यहाँ पर डायरेक्टली प्रॉपर नाम दिए हैं. अगर इन डिटेल मीडिया फॉर्मेशन देखिए, मीडिया कंपोनेंट्स देखिए तो लैक्टोज यूज़ किया जाता है, कैल्शियम कार्बोनेट यूज़ किया जाता है, कॉन्स्ट्रिप्लिकर, ग्लूकोज, फिनिल एसिटिक एसिड, सोडियम हाइड्रोजन फॉस्फेट और कुछ एंटीफोम एजेंट्स यूज़ किए जाते हैं. अपार्ट फ्रॉम दिस वी यूज स्मॉल अमाउंट ऑफ ट्रेस एलिमेंट्स लाइक कॉपर, मैग्नीज and cobalt, or we use the growth factors like vitamins and amino acids, and some precursors are also being used to enhance the growth of the penicillin. Okay, so that is all about the penicillin. Some conditions are required to maintain for the growth of the penicillin, like penicillin ये एक एरोबिक ऑर्गेनिज्म है. सॉरी, मैं आपको बताना भूल गई कि व्हाट आर द प्रोड्यूसर ऑर्गेनिज्म फॉर द प्रोडक्शन ऑफ दिस पेनिसिलिन. पेनिसिलिन इज प्रोड्यूस्ड बाय द फंजाई पेनिसिलिन नोटाटम एंड पेनिसिलिन क्राइसोजिनम. ये दो मोस्ट वाइडली यूज्ड फंजाई है एट द इंडस्ट्रियल लेवल फॉर द प्रोडक्शन ऑफ पेनिसिलिन. If we will talk about Penicillin notatum, it has high efficiency for the production of antibiotic. Sometimes we use both Penicillin notatum as well as Penicillin chrysogenum. Means we transfer the gene of Penicillin notatum to the Penicillin chrysogenum to enhance the production. Okay, so these both fungi are being widely used for this production. The specific conditions which are required, that is penicillin ये एक aerobic fungi है, aerobic organism है, तो oxygen supply is very critical and it is must. Optimum pH for penicillin growth is 6 to 6.5. Temperature is 20 to 25 degree Celsius. तो ये conditions maintain करना बहुत ज़रूरी है while production of penicillin. Okay, so this media is been used for the production of penicillin. Now the most important factor for the production is to maintain the culture or to maintain the starter culture. तो आप क्या करेंगे? To start the fermentation process, a number of spores are introduced. Number of spores, penicillium notatum के spores introduced किए जाते हैं into the small flask, conical flask of 250 to 500 ml. That flask is allowed for the incubation for 3 to 4 days. And now at this stage what happens? There is unstable growth or there is mat growth of that spores, and in that condition आपको pH और temperature, oxygen ये सब maintain करना बहुत ज़रूरी है. तो वो 250-500 ml के फ्लास्क में ही एजिटेटर सिस्टम प्रोवाइड की जाती है, क्योंकि पेनिसिलिन ये एरोबिक ऑर्गेनिज्म है, तो वो अपनी ग्रोथ तभी दिखाएगा जब उसे ऑक्सीजन का सप्लाई मिलता है. ठीक है, तो फिर उस फ्लास्क में प्रॉपर एजिटेटर सिस्टम प्रोवाइड की जाती है and then it is incubated for 5 to 7 days. means up till the growth आपको मिलती नहीं है, दिखती नहीं है, तब तक आप उसे incubate कर सकते हैं. तो इस incubation के समय में होता क्या है? at this stage spores become active. ओके, जो हमने स्पोर्स इनोकुलेट किए थे वो स्पोर्स एक्टिव होते हैं, एंड देन देयर इज फॉर्मेशन ऑफ द vegetative cells. Okay, spores convert into the vegetative cells. Overall conditions सब maintain की जाती है, parameters maintain किए जाते हैं, pH, temperature, agitation provide किया जाता है. So these conditions are being maintained. After the mat growth of the mycelium, ये जो culture है ओके, वो कल्चर ट्रांसफ़र किया जाता है इनटू द फर्मेंटर टैंक, वो रहेगा, वो होता है आपका इनोक्यूलम. ओके, सो दैट इज़ द इनोक्यूलम, जो पहली स्टेप यहाँ पर दिखाई है इनोक्यूलम. सो दैट इनोक्यूलम इज एडेड इनटू द फर्मेंटर टैंक along with the media which I have told you earlier, and then again the conditions are being maintained like aeration, temperature, pH, and antifoams are being added. So plus again it is allowed to ferment for a certain time and at a certain temperature. After the process of fermentation is been completed, the product is been recovered, the product is been processed. Okay, so how the recovery is been done, द मीडियम जो हमने यूज़ किया था वो मीडियम दिखाया है, बैच फर्मेंटर यूज़ किया गया है. ओके, मीन्स आफ्टर वन बैच इज कम्प्लीटेड द सेकेंड बैच इज बीन एडेड, और यहाँ पर जो न्यूट्रिएंट्स हम ऐड करते हैं वो फेड बैच की तरह ऐड करते हैं, which I have told you earlier. Means here substrate is added in the small amount at various times, at various time interval in the fermentation, and then the volume has been increased. ठीक है, तो वो मीडियम ऐड किया जाता है बैच फर्मेंटर में. और जो स्टार्टर कल्चर है पेनिसिलिन का, मतलब जो अभी हमने उनका the process which I have told you now, that is the starter culture to increase the प्रोडक्शन ऑफ द माइसेलियम, और टू इंक्रीज द, और टू मेड, जो इनोकुलम हमने प्रिपेयर किया है वो हम डालेंगे अलॉन्ग विद द मीडिया, एंड देन फर्मेंटेशन इज बीन कैरिड आउट. ओके, एंड नाउ how your processed or how your product is been processed, how your product is been recovered, that is been shown. रोटेटिंग फिल्टर यूज़ किया जाता है, स्पेशली पेनिसिलिन को प्यूरिफाई करने के लिए रोटेटिंग फिल्टर यूज़ किया जाता है. द फिल्ट्रेट इज बीन कलेक्टेड, वो फिल्ट्रेट को डिसॉल्व किया जाता है इन द ब्यूटिल एसीटेट. ओके, ब्यूटिल एसीटेट क्या करता है, इट फॉर्म्स द पोटेशियम, it forms the precipitate. Okay, so then afterwards potassium ions are been added to precipitate the salt of penicillin. हमें penicillin चाहिए, so it is important to precipitate out the penicillin. So penicillin is precipitated by adding the potassium ion along with the butyl acetate. जो प्रिसिपिटेट हमें मिलता है उसे वॉश किया जाता है, फिल्टर किया जाता है, ड्राई किया जाता है. तो वो जो ड्राई होने के बाद वी गेट इट इन द पाउडर फॉर्म. 99.5% प्योर पेनिसिलिन हमें मिलता है, नॉट कंप्लीट 100% बट 99.5% पेनिसिलिन वी गेट, एंड देन इट इज़ यूज्ड एज there is modification, chemical और enzymatic modification होता है to make the penicillin antibiotic. So in this way we get the penicillin antibiotic from the fungi Penicillium notatum and Penicillium chrysogenum. The very हर जो भी अभी तक से हमने प्रोडक्शन मेथड्स देखे हैं, तो उसमें स्टार्टर कल्चर और इनोकुलम प्रिपरेशन बहुत इम्पॉर्टेंट है. इनोकुलम की ग्रोथ बढ़ाई जाती है, और फिर ग्रोथ बढ़ाने के बाद इट इज़ एडेड इनटू द फर्मेंटर टैंक अलॉन्ग विद द मीडिया. पैरामीटर्स कंट्रोल किए जाते हैं, फर्मेंटेशन प्रोसेस कैरी आउट की जाती है इन अ सर्टन टेम्परेचर एंड एट द सर्टन टाइम, and afterwards the process has been or the product is been recovered. So this is all about the we can say production of the antibiotics. Thank you.